first step in infection of a cell is attachment or adsorption. The virus will attach to the cell surface via ionic interaction, which are temperature independent. The viral attachment protein recognize specific receptor may be protein, carbohydrate, or lipid on outside of the cell. The cell without appropriate receptor are not susceptible to virus. Second step is penetration. The virus will enter the cell in variety ways according to the nature of the virus. For the envelope virus, there are two mechanisms. First, entry by fusing with plasma membrane. The virus will fuse directly with plasma membrane, the internal component of virion immediately delivered to the cytoplasm of the cell. Second entry is via endosome at cell surface. Some viruses require acid pH for fusion because they are unable to fuse directly with the plasma membrane. The virus will taken up by invagination of the membrane into endosome. As the endosome becomes acidified, latent fusion activity of virus protein become activated by the fall in pH and the virion membrane fuse with the endosome membrane. The result is in delivery of internal components of virus to cytoplasm of the cell. For the non-enveloped virus, they may cross plasma membrane directly or may be taken down into endosome. They then cross or destroy the endosomal membrane. The third step is uncoating. Uncoating is the release of the viral genome from its protective capsid to enable the viral nucleic acid to replicate. The nucleic acid must sufficiently uncoated that virus replication can begin at this stage. When the nucleic acid uncoated, infectious virus particle can't be recovered from the cell, and it starts off eclipse phase. It will last until new infectious variants are made. Next is the transcription and translation of the viral genome replication. It is the synthesis of viral nucleic acid and protein. First, transcription will occur, which is synthesis of mRNA. Second is the translation process, which is viral mRNA is translated on cell ribosome into structural and non-structural protein. For the DNA virus, it will utilize the cell's transcriptional mRNA and translational machinery, which is RNA polymerase III. Some will alter the cell machinery for their own use. Next, is the maturation or assembly process. The new virus genomes and protein will assemble to form new virus particle. The new virus particle are assembled may be maturation steps that follow the initial assembly process. The last process is release the virus. The enveloped virus will release by budding, but budding not necessarily will kill the cell. Some may be able to set up persistent infections. Naked virus will release by rupture of infected cells. Bear in mind that not all released viral particles are infectious. The ratio of non-infectious particle varies with the virus and growth conditions.